We will start in just a couple of minutes. I will start by sharing my slides. Welcome to the final live session of the course Structural Biology. This will be a summary session where I will quickly go through some of the main points of all the lectures. To start with, we will be talking about biological macromolecules. As you know, there are four major classes of biological macromolecules, in which the first is carbohydrates. Now, carbohydrates serve as the immediate source of energy of living organisms. The general formula of monosaccharides is CH2O whole N. And that is why they are also called carbohydrate, since they have carbon and hydrate meaning water. Monosaccharides are the simplest form of carbohydrates, and as such, they are considered to be their building blocks. So you can see this is the structure of glucose, fructose, which is a 5-membered sugar, and galactose. These are monosaccharides. Because monosaccharides have many OH groups, they can be joined to one another by covalent bonds called glycosidic bonds. So two monosaccharides come together and they lose a molecule of water. That is, a dehydration reaction takes place to form a glycosidic bond and a disaccharide. So you can see sucrose is made up of one molecule of glucose and one molecule of fructose. Similarly, lactose is made up of one molecule of galactose and one molecule of glucose. Disaccharides are classified based on the location of carbonyl groups and the number of carbons. And in polysaccharides, numerous different monosaccharides come together forming glycosidic bonds. Like you can see, this is a polysaccharide glycogen made up of numerous number of glucose units. The second kind of biological macromolecule is called fat. In fats, basically, there is a glycerol molecule which has 3 OH groups, and there are fatty acids attached to each OH group of glycerol. Fatty acids are basically have a carboxylic acid group and a long aliphatic side chain. So an esterification reaction takes place where a water molecule is lost and there are three ester bonds formed which connect each of the three fatty acids to the OH of glycerol. Now you can have a saturated fatty acid in which the aliphatic chain has only single bonds, or you can have an unsaturated fatty acid where the aliphatic chain has a double bond. The double bond introduces a kink or bend in the molecule. And in phospholipids, what happens is that two of the OH of glycerol are attached to fatty acids, which can be saturated or unsaturated, but one OH is attached to a phosphate group. Because of the negative charge of the phosphate group, this is called this portion becomes hydrophilic and it is called the head, whereas the aliphatic chains form the hydrophobic tails. Steroids are lipids characterized by a carbon skeleton consisting of 4 fused rings. So there are 4 rings A, B, C, D here, and this is an example of the steroid cholesterol. The third kind of biological macromolecule is called a nucleic acid. There are 2 main classes of nucleic acids, deoxyribonucleic acid or DNA and ribonucleic acid or RNA. Nucleic acids are polynucleotides, that is, long chain-like molecules composed of a series of nearly identical building blocks called nucleotides. Each nucleotide consists of a nitrogen-containing aromatic base. You can see there are 4 examples of the aromatic bases shown here: adenine and guanine, which consist of 2 fused rings, and cytosine and thymine, which consist of a single ring. Other than the nitrogenous base, the nucleotide consists of a pentose or 5-membered sugar ring, which is attached to a phosphate. Each nucleic acid consists 4 of 5 possible nitrogen-containing bases: adenine, guanine, cytosine, and thymine. And in RNA, you have uracil. All the nucleic acids contain the bases AC and GT, which are found in DNA, while U is found in RNA. And as you know, adenine and guanine, between them there is complementary base, between adenine and thymine, there is complementary base pairing and 2 hydrogen bonds, whereas between guanine and cytosine, there is complementary base pairing with 3 hydrogen bonds. The fourth kind of biological macromolecule is proteins. Now, protein consists of the building block called amino acid, which basically there is a central alpha carbon to which there is an NH2 group, a COH group, a H group, and an R group, which can be of 20 different types. Now we'll quickly go through the 20 naturally occurring amino acids. If the R group is H, then it is glycine. If the R group is CH3, it is alanine. Now it is important to remember the 3-letter and 1-letter code associated with each amino acid. The R group can be nucleophilic, like CH2OH in case of serine, and if a methyl group is added to the alpha position of the alcohol, then it forms a threonine. And instead of OH, if it is SH, then it is a cysteine. Now the R group can also be hydrophobic. Like if it is an isopropyl group, it forms valine. If it is a butydyl group, it forms leucine. If it is an isobutydyl group, it forms isoleucine. Methionine is basically a CH2CH2SCH3. So if you are asked about the 2 sulfur-containing amino acids, it is cysteine and methionine. Now, proline is very unique because it is a cyclic amino acid. Now, if the R group is aromatic, like a phenyl ring, then it is called a phenylalanine. If an OH is added to the para position of phenylalanine, it forms tyrosine. If the R group has an indole ring, it forms tryptophan. Now, the R group can also be acidic. If it is CH2COOH, it forms an aspartic acid. If an additional CH2 is inserted, then it is glutamic acid. The R group can also be an amide. If it is CH2CONH2, then it is asparagine. If an additional CH2 is attached, then it forms glutamine. And finally, there are three kinds of basic R groups. Firstly, and if it is an imidazole ring, that is histidine. If it is 4CH2 followed by NH2, that is lysine. And if it is 3CH2 followed by a guanidinium group, that is arginine. Next, we will be talking about the isoelectric point of amino acids. Now, as you know, in neutral environment, amino acids exist as a zwittered ion. That is, the carboxylic acid group is deprotonated and the proton is accepted by the amine group. So due to the presence of a single positive and a single negative charge, the net charge is 0. So isoelectric point is the pH at which the amino acid exists in this neutral zwittered ionic form. Now, if you have an alkaline pH, this H plus is also lost and the molecule gets a net negative charge. Or if you have an acidic pH, then the deprotonated COO minus gains an H plus and the net charge becomes positive. Isoelectric point is generally found by taking an average of the two pKa's on either side of the zwittered ionic form. So if we want to, this was the example of glycine, in which the side chain is a simple H atom. Now, if you have an aspartic acid, then the neutral form or zwittered ionic form is when the side chain is protonated, but the main chain carboxylic H is lost and accepted by the NH2. So this is the neutral charge or zero charge zwittered ion. Now at pKa 2.1, which is acidic conditions, the main chain CO minus gains an H and the net molecule gets a net +1 charge. At pK around 3.9, the side chain COH is lost and the molecule gets a net negative charge. If we increase the pKa further to 9.8, then the main chain NH3 plus also loses the proton and the molecule gets a net -2 charge. Now, if we want to find the isoelectric point or the pH at which the zwittered ion exists, we need to take the average of the two pKa on either side of the zwittered ionic form. So the average of 2.1 and 3.9, which will give us 3. Similarly, if we take the average for basic amino acids like lysine, if we take the average of the side chain NH2 and the main chain NH2, then we will get the PI for a basic amino acid like lysine. Now we will talk about protein architecture. So like we saw, amino acids are the building blocks of proteins. Two amino acids can come together and lose a molecule of water, that is from the COOH group an OH is lost, and from the NH2 group an H is lost. Losing this molecule of water or a dehydration reaction, a peptide bond is formed, which is basically a C double bond O NH bond. And this is a partial double bond because the lone pair of nitrogen gets delocalized into the carbonyl and the peptide bond is a partial double bond. Then comes the secondary structure, where basically there is interpeptide hydrogen bonding to form structures like the alpha helix or beta sheet. Now in alpha helix, there is 4-to-1 hydrogen bonding. That is the I plus 4th amino acid hydrogen bonds with the ith amino acid. And in beta sheet, there is hydrogen bonding between two adjacent beta sheets. In tertiary structure, the secondary structures come together due to folding and side chain interactions to form a tertiary structure. So basically, you can have alpha helices and beta sheets held together by side chain interactions. The final level is the quaternary structure, where more than one tertiary structure is held together by different hydrophobic or the van der Waals interactions. So quaternary structure is the final level of this protein architecture. Now, instead of tertiary structure, it is sometimes called super secondary structure. And the examples of these are if we have a bundle of 4 helices represented by 4 alpha symbols, that is a 4 helical bundle super secondary structure. Or there can be alternate beta and alpha folds, which is called a Rossmann fold, and it is also an example of a super secondary structure. Next, we will look at the torsional angles and the Ramachandran map. So here the peptide plane is shown. And if we want to find the phi torsional angle, then we have to start from one of the carbonyls, carbonyl carbon, go to the adjacent nitrogen, alpha carbon, and the next carbonyl carbon. So the dihedral angle formed by these 4 atoms is the phi dihedral angle. In case of the psi dihedral angle, we will start from the amide NH, go to the adjacent C alpha, then carbonyl carbon, and the next amide NH. So from one end to another, the torsional angle that is there is called the psi torsional angle. In case of the omega torsional angle, you have to start from one of the C alphas, go to the carbonyl carbon, amide NH, and then the next C alpha. So the torsional angle formed between two adjacent C alphas is the omega torsional angle. This can be 180 degrees as shown here, and that is called an S-trans conformation, or it can be 0 degrees as shown here, and that is called an S-cis conformation. The chi dihedral angle is the dihedral angle associated with the side chains. Now, some combinations of torsional angles are much more likely than others, and this is shown by the Ramachandran plot, where in the x-axis you have phi and in the y-axis you have psi. So the white regions of the Ramachandran plot are the disallowed phi-psi angles, which are disallowed due to steric repulsions. The red regions are the ones which are completely allowed. Like you can see, this particular red region represents the alpha helical conformations, while at the top this region is the beta sheet conformations. The yellow regions represent border space, meaning those phi-psi which are partially allowed. That is allowed in some cases and disallowed in others. This gives rise to this portions, which is a left-handed alpha helix or alpha L conformation. So there are many different kinds of bonds that can stabilize a protein. The van der Waals forces contribute about 0.1 kilocalorie per mole. The hydrophobic bond has an energy of 1 kilocalorie per mole, similar to the hydrogen bond, which also has 1 kilocalorie per mole. The ionic bond energy can be up to 3 kilocalorie per mole, and the strongest is the covalent bond up to 90 kilocalorie per mole. The weak forces, that is the forces other than the covalent bond, are constantly forming and breaking under physiological conditions, and they influence the structure and behavior of all biological molecules. There is another kind of force called the disulfide bond, where basically 2 cysteines come together and they lose 1 hydrogen each, and a single bond forms between the 2 sulfurs called the disulfide bond. Disulfide bonds increase the thermal stability of a protein. And so we to introduce such a bond, we have to first identify the region where it can be introduced without any steric clashes. Now, protein folding is the physical process by which a protein chain acquires its native 3D structure, a conformation that is usually biologically functional in an expeditious and reproducible manner. Now, folding of a globular protein is thermodynamically favored, so the delta G must be negative. The folding process involves going from a multitude of random coil conformations to a single folded structure. So basically, if this is the energy of folding, then you can have many unfolded states, but the native state is a single conformation, which is usually assumed to be the lowest energy conformation. The folding process involves a decrease in randomness and thus a decrease in entropy. That is, the delta S is negative because it is going from several unfolded states which can move in different directions to a single folded state which is fixed by different intramolecular forces. So delta S is negative and it has a positive contribution to delta G. Basically, the formula of delta G is delta H minus T delta S. Since delta S is negative, this whole term T delta S becomes positive and has a positive contribution to delta G. This decrease in entropy is termed as conformational entropy. An overall negative delta G is needed in order for the protein folding to be favorable, and that is possible if the net delta H is negative. So if you look at the energy diagram, basically you can see that T delta S, which is represented by this, is the decrease in conformational entropy on folding, and that is unfavorable. That in other words, the T delta S term is overall negative. It becomes positive due to the minus sign. Now, there is something called hydrophobic effect, which is basically upon protein folding, hydrophobic residues move to the interior of the protein and caged water molecules are released. So due to the hydrophobic effect, there is a favorable contribution to entropy. So the net entropy, which is unfavorable from conformational entropy and favorable from hydrophobic effect, the net entropy has a negative contribution, which due to the minus sign becomes positive, that is unfavorable. The net delta H for folding is favorable, that is, it has a negative conformation, and this delta H comes from the formation of secondary structure where there are hydrogen bonding interactions, van der Waals interactions, or dipole-dipole interactions. There is also contribution from formation of the tertiary structure, but that is slightly unfavorable. So the overall enthalpy of folding is favorable by 5 to 15 kilocalories per mole. And overall, this delta G, which is delta H minus T delta S, is slightly favorable, and that makes the protein folding a spontaneous process. Now, this is the electromagnetic spectra, and you can remember, you have to remember the wavelength, and having an idea of the wavelength will give you an idea of the energy. So the gamma rays, 1 second. So as you know, energy is h nu, or it can be written as h c by lambda. So if you know the lambda, you can have an idea of the energy. So the shortest wavelength are possessed by the gamma rays. That is, they have the highest energy. Then the X-rays, then ultraviolet, visible, IR or infrared, microwave, and radio waves have the largest wavelength, that is the smallest energy. So the macromolecules like proteins cannot be measured by visible light because their size is much smaller than the wavelength of the visible light. Instead, biological macromolecules are usually studied by X-rays. So one kind of point to be remembered is that for an object to be visible under magnification, the wavelength of the light must be, roughly speaking, no larger than the object. Now we will briefly look into the concept of recombinant DNA. So DNA cloning is the process of making multiple identical copies of a particular piece of DNA. In a typical DNA cloning procedure, the gene or other DNA fragment of interest is first inserted into a circular piece of DNA called a plasmid. So this is basically a plasmid. It is digested with restriction enzyme. What means is that a part of the plasmid is cut out and the target gene is inserted over there using DNA ligase. And this forms the recombinant plasmid. Next, the recombinant plasmid is introduced into bacteria, and the bacteria carrying this plasmid are selected and grown. As these bacteria reproduce, they make copies of the plasmid, and from a small amount of the initial DNA, a large amount of the recombinant DNA can be formed. So what is the point of making so many copies of the DNA sequence in a plasmid? This is because sometimes we need lots of DNA to conduct experiments or build new plasmids. In other cases, the DNA encodes a useful protein, and the bacteria can be used as factories to make the protein. For example, human insulin gene is expressed in E. coli bacteria to make insulin, which is used by diabetics. Now we will look into polymerase chain reaction. So polymerase chain reaction has three major parts. First is denaturation. In this, the two strands of DNA are separated, and it is accomplished by heating the starting material to temperatures of above 95 degrees Celsius. Each strand is now a template on which a new strand can be built. The next step is called annealing. In annealing, the temperature is reduced to about 55 degrees Celsius so that a primer can anneal to DNA. So primer is a small fragment of DNA, and it is needed because DNA polymerase can only add a nucleotide if there is a pre-existing 3' OH group. So in order for this 3' OH group to exist, an initial primer needs to be annealed to the DNA template. The third step is called extension, and here the temperature is raised to about 72 degrees Celsius, and the DNA polymerase begins adding nucleotides one by one at the end of this annealed primer, and the nucleotides are added following the complementary base pairing rules. And this whole process is called polymerase chain reaction. So if you start with the DNA and you carry out n cycles of the polymerase chain reaction, you will end up with 2 to the power n copies of the DNA. In Sanger sequence, what happens is that along with the deoxyribonucleotides or DNTP, a number of chain terminating dideoxyribonucleotides or DDNTP is also mixed into the polymerase chain reaction mixture. In DDNTP, basically this 3' OH is not present. So whenever a DDNTP is incorporated in the extension step, the extension step immediately stops. because there is no OH for adding further nucleotides. So what happens is in the PCR, we get a mixture of truncated DNA sequences terminated at random lengths by this DDNT. Then these truncated sequences can be separated by capillary gel electrophoresis, and using a laser excitation, these truncated sequences can be identified. And this is used to find the sequence of the entire DNA, and it is called Sanger sequencing. There are several ways of purifying proteins. One is the gel filtration chromatography, and the principle is the separation by size. So the column beads have different size pores. As the column flows, the large proteins remain outside the pores and are therefore rapidly eluted, while the small proteins shown by the red dots here enter the pores and they flow slowly. So the large molecules will be eluted first and the small molecules will be eluted later. Next, we have ion exchange chromatography, and the principle is separation by charge. So the beads have charged group. So if the charged group in the stationary bead is positive, it will bind negative, that is acidic amino acids. And if the charged beads are negative, they will bind positive groups like basic amino acid. So different proteins will bind with different affinity, and then they can be eluted with increasing amount of salt. So in this particular example that is shown, the immobilized surface is negatively charged. So the positively charged protein will stick while the negatively charged protein will come out. Then if the salt is slowly added, then the positively charged protein will also come out later. So here the separation is based on charge. There are several zones during the crystallization of proteins. So firstly, the solubility line, which is the dark blue line, divides the diagram into an undersaturated, that is this white background, and supersaturated zone. This entire area is the supersaturated zone. The undersaturated zone represents a single phase and stable solution where the protein molecules shown by these blue circles are in solution. With increasing saturation, the solubility line, that is this dark blue line, is crossed and the solution becomes supersaturated. The supersaturated region is divided into a metastable zone and an unstable zone, which is this red region. The metastable zone again consists of a crystal growth zone, which is this light blue zone, and a nucleation zone, which is this dark blue zone. In the crystal growth zone, only transient nuclei are formed that do not reach a critical size. If the supersaturation proceeds, then from the crystal growth zone, you enter the nucleation zone, where nuclei can achieve a critical size and become stable. As the nuclei become larger and crystals start to appear, the protein concentration in the solution is decreased and the system reaches the crystal growth zone again from the nucleation zone. In this zone, the crystals continue to grow at low supersaturation. When the supersaturation is too high, it reaches the unstable zone, where the proteins begin to precipitate out amorphously. Now we will talk about how X-rays are produced. So basically this setup is called a cathode ray tube, and there is a filament through which a current is passed. This current heats up this filament, and when it is hot enough, electrons are emitted, which is called thermionic emission. These electrons are accelerated through a high potential difference of 50 to 100 kilovolts through a metal target or anode which is positively charged. Because it is positively charged, it is able to accelerate the negatively charged electrons. This anode is usually made of tungsten, and it attracts the negatively charged electrons because it is positively charged. The electrons then collide with the anode and they rapidly lose their acceleration by losing kinetic energy. And this kinetic energy that is lost forms the X-rays. So the electron beam is focused from the cathode to the anode by something called a focusing curve. And the distance between the filament from where the electrons are being produced to the X-ray tube target is only 1 centimeter. And the velocity of the electrons while they are being accelerated reach from 0 to half the speed of light. The projectile electron interacts with the orbital electron of the target atom, resulting in the conversion of electron kinetic energy into thermal energy, that is heat, and electromagnetic energy in the form of infrared radiation, which is also heat. Only 1% of the energy is released as X-rays. Just give me 1 minute. Now we will be talking about Bragg's law of diffraction. So basically, if this is a lattice of a crystal, if the ray 1 strikes the top layer of the lattice, it will bounce off or get reflected, and this is the reflected ray 1. Here, since we are studying diffraction, it is diffracted beam. The ray 2 will get diffracted from the lower layer of the lattice to form the other part of the diffracted beam. Now, we know that we can see that the part difference between ray 1 and ray 2 is basically BC plus BD. So B is basically here. 1 second. This is B. So BC plus BD is the path difference between ray 1 and ray 2. And if we consider the interplane distance to be small d, then small d sine theta is the BC length, and it is also the BD length. So basically the difference in path between ray 1 and ray 2 is 2d sine theta. The condition for constructive interference is that this path difference is an integral multiple of the wavelength lambda. So what does this mean? Constructive interference occurs when two waves have their crest at the same position and their trough at the same position. Then the amplitude of the net wave doubles, and it is called constructive interference. Now, if the phase of the two waves are opposite, like one has a crest while the other has a trough, and when the first has a trough, the second has a crest, then the two waves completely cancel out, and it is called a destructive interference. Now, if the path difference between these two waves is an integral multiple of the wavelength, then there will be constructive interference. So this n is called, can be 1, 2, 3 different integers, and it is called, if it is 1, it is called first order diffraction. If it is 2, second order diffraction, and so on. So the path difference is 2d sine theta is n lambda. And this is the Bragg's law of X-ray diffraction. Here n is called the order of reflection, d is the spacing between two lattice planes, and theta is called the Bragg angle. There are certain points that must be remembered while considering the geometry of Bragg's law. Firstly, the incident beam, the normal to the reflection plane shown by this dotted line, and the diffracted beam are always coplanar, that is, they are in the same plane. The angle between the diffracted beam and the transmitted beam is always 2 theta. So this particular angle is always 2 theta. Since sine theta cannot be more than unity, so sine theta is generally less than unity. So n lambda is less than 2d. For n equal to 1, lambda is less than 2d. So the wavelength of light that we take should be less than twice the spacing between the crystal plane. Now we will talk about Bravais lattice. So a lattice is a set of points constructed by translating a single point in discrete steps by a set of basis vectors. A Bravais lattice is an infinite array of discrete points with identical environment. In 3 dimensions, there are 14 unique Bravais lattice in 3 dimensions. And these belong to 7 crystal systems. So the first crystal system is called cubic. It can be a simple cubic where there is an atom at each of the vertex of the cube, or it can be body-centered cubic. That is, there is an atom at the vertex and one atom in the middle of the cube. Or it can be face-centered cubic. That is, there is one atom at each of the vertices and one atom at the middle of each of the 6 faces. The second crystal, so in cubic, the characteristic is that all the sides are equal, that is A equal to B equal to C, and the angles between the X, Y, Y, Z, and so on axis, alpha, beta, gamma, are all 90. Now in tetragonal, alpha, beta, gamma are all 90, but 2 sides are equal and the third side is not equal. Tetragonal can again be simple tetragonal or body-centered tetragonal. Next is orthorhombic, where all the sides have different lengths, but all the angles are equal and they are 90. And they can be simple, body-centered, face-centered, or base-centered. Base-centered basically means there is an atom at each of the vertices and one atom each on the opposing planes, on one pair of opposing planes. Then is monoclinic, where all the sides are of different lengths and 2 angles are equal and equal to 90, but one angle is different. Monoclinic can be simple monoclinic or base-centered monoclinic. The 3 final crystal systems have only one Bravais lattice each. One is rhombohedral, where the sides are all equal and the angles are also equal and equal to 90 degrees. Then is hexagonal, where 2 sides are equal, but the third is unequal, and 2 angles are equal to 90 degrees, and the third is 120 degrees. And finally, the least symmetric crystal system is called triclinic, where all the sides are unequal and all the angles are unequal, and none of them are equal to 90 degrees. So if you add up, there are 14 Bravais lattices shown here, and there are 7 crystal systems, which are basically cubic, tetragonal, orthorhombic, monoclinic, rhombohedral, hexagonal, and triclinic. Now we will talk about Miller indices. So crystal lattice may be regarded as made up of an infinite set of parallel equidistant planes passing through the lattice points, and these are known as lattice planes. The orientation of the planes or faces in a crystal can be described in terms of their intercepts on the 3 axes. Miller introduced a system called Miller indices to designate these planes. He introduced a set of integers HKL to specify a plane, and this set of integers is called Miller indices. There is a certain procedure to find Miller indices. So suppose we take this as the example. The first step is to find the intercept along the XYZ axis in terms of the lattice constant A, B, and C. So the intercept along this x-axis is 3. So you have written 3. Along y-axis it is 2, and along z-axis it is also 2. The second step is to find the reciprocal of these intercepts. So that becomes 1 by 3, 1 by 2, and 1 by 2. The third step is to find and multiply each of these reciprocals by the least common denominator. So the least common denominator of 3 and 2 is 6. So if you multiply 1 by 3 by 6, it becomes 2, and 1 by 2 by 6, it will become 3. And the final step is that these integers are written in brackets like this. So first brackets, and they are separated by, you can put commas or you cannot put commas, but you put the integers in first brackets. So the Millard indices of this plane is 2, 3, 3. Now we will look at the structure factor and diffraction pattern. So the maxima of electron density function, denoted as rho xyz, shows the location of the atoms. That is the internal structure of the crystal. It is part of the real space. Now, you can Fourier transform this to find the reciprocal space, which is basically made up of mod of capital F HKL, which is called the structure factor, and it represents the amplitude of the wave. The other information that is obtained from this reciprocal reciprocal space is the phase information, that is phi HKL. So in a wave, the structure factor, the amplitude is the distance from the base to the crest, or the base to the trough, while the phase information is the horizontal shift of the wave. So if we look at the structure factor in terms of a vector, the magnitude of the vector is the amplitude, and the angle of the vector from the x-axis is the phase information. Now we will talk about NMR active and NMR inactive nuclei. So parallel pairing results in addition of the spin values, and antiparallel pairing cancels out the spin of the particles. The principle of NMR spectroscopy is based on the nuclei having a net spin value which is non-zero. So there are certain rules. Like if the atomic mass and the atomic number are both odd, like in case of 1H, so the atomic mass is 1 and it is odd. The atomic number is also 1 and it is odd. That nucleus is NMR active. If the atom is like 13C, where the atomic mass is odd, that is 13, and the atomic number is 6, that is even, that is also NMR active. So we have 1H NMR spectroscopy and 13C NMR spectroscopy. Now, if the atomic mass is even, like in deuterium, the atomic mass is 2, and the atomic number is odd, like in deuterium, the atomic number is 1. That is also NMR active. The only case where the nuclei is NMR inactive is if both the atomic mass and atomic number are even, like in case of 12C, where the atomic mass is 12 and the atomic number is 6. So using these rules, you can find out whether an NMR nucleus is NMR active or NMR inactive. We will briefly look at different examples of NMR spectra. So in CH3Br, there is a single type of hydrogen, which is this CH3 hydrogen, and so a single peak is observed. The there is no point in finding the integral of this peak, because integral only makes sense when we have two or more different kinds of protons. Now, in case of acetone, again, there is a single kind of proton, which is the CH3, and so a single peak is seen. And this is the same case for tertiary butyl bromide. There is a single kind of proton, since all the CH3 protons are equivalent, so a single peak is seen. Now, if you have this molecule, there are two kinds of protons. One is directly attached to the carbonyl group, shown in blue, and one is directly attached to oxygen, shown in green. So two different peaks are observed. Now, compared to the blue hydrogens, the green hydrogens are more de-shielded, since it is directly attached to the electronegative oxygen. So the ppm value of the green hydrogen is higher. If you integrate the two peaks, the integrations are 1:1, since both kinds of hydrogens have an equal number of hydrogen, that is 3 hydrogens each. Now, if we look at the splitting of peaks due to spin-spin coupling, we can see that in ethyl bromide, there are two kinds of hydrogens. So there are two peaks shown by blue and green. Now, the green CH2 will be more de-shielded, since it is directly attached to the electronegative bromine. So the green CH2 has higher ppm values. Now, the green CH2 has 3 equivalent CH3 attached to it. So the rule of splitting, as you know, is n+1. So since there are 3 hydrogens adjacent to it, the green peak will split by 3+1. That is, it will have 4, it will split into 4 peaks. Now the blue peak has 2 hydrogens adjacent to it. So it will split into 2+1, that is 3 peaks. Now if we look at propyl bromide, there are 3 kinds of hydrogens. So there are 3 main peaks. The blue peak will be most de-shielded because it is directly attached to the bromine. Then the black peak, and finally the green peak. Now the blue peak has 2 hydrogens adjacent to it, so it will split into 3 peaks. The black peak has 3 + 2, 5 hydrogens adjacent to it, so it will split into 6 peaks. And finally, this green peak has 2 hydrogens adjacent to it, so it will split into 3 peaks, or a triplet. Now let's look at Lambert-Beer's law. So Lambert-Beer's law is essentially absorbance equal to ALC, where A is the molar absorptivity or extinction coefficient, and it is a wavelength dependent constant for the species being analyzed. L is the path length in centimeter, and it is usually the diameter of the cuvette or sample holder. So it is the same if the same cuvette is used. C is the concentration of the sample in mole per liter. So basically, if you plot absorbance with concentration, then a straight line is observed with intercept at 0 and slope equal to A into L. Now, if we look at UV visible spectroscopy in proteins, most proteins have an absorbance maxima at 280 nanometer compared to nucleic acids, which absorb at 260 nanometer. So, in order to find the concentration of a protein in mg per ml, this formula is used, which is 1.55 into the absorbance at 280 nanometer, which is caused by proteins, minus 0.76 into the absorbance at 260 nanometer, which is caused by nucleic acid. Now, the basis for UV-vis spectroscopy in proteins is the presence of aromatic amino acids like phenylalanine, tyrosine, or tryptophan. So proteins which lack these residues cannot be observed by UV visible spectroscopy. CD spectra of proteins is another kind of spectroscopy which helps in distinguishing between the protein secondary structures. So generally, each secondary structure has a characteristic CD spectra. So if it is an alpha helix, it will have two peaks towards the negative value of molar ellipticity. That is at 208 nanometer and 222 nanometer, it will have the negative peak, and around 192 nanometer, it will have a positive peak. The characteristic of beta sheet is a positive peak around 195 nanometer and a negative peak around 218 or 216 nanometer. And for random coil, the characteristic is a negative peak around 195 to 200 nanometer. So if there is a polylysine, looking at the CD spectra, we can find out what is the secondary structure adopted by the polylysine. Then we will talk about green fluorescent protein. So in green fluorescent protein, the chromophore is essentially a serine tyrosine glycine tripeptide. So this tripeptide, there is basically an autocatalytic cyclization, and the final fluorophore has a lot of conjugated double bonds, due to which we can see the green fluorescent color. There can be several mutants. One is, instead of green fluorescence, it shows blue fluorescence. So there what happens, instead of serine tyrosine glycine, you have serine histidine glycine. So the tyrosine has been mutated to histidine. Now, EGFP is another mutant where it is enhanced green fluorescence. So the intensity of the green fluorescence is considerably increased. And here, the serine at 65 is mutated to a threonine. Then you can have a cyan colored fluorescence. And here, there are two mutations. The serine at 65 is mutated to threonine, and the tyrosine at 66 is mutated to tryptophan. And finally, you can have yellow fluorescence, and there again there are two mutations. There is one mutation where the serine 65 is mutated to a glycine 65. So these are the four mutants of green fluorescence protein that have biological significance. So we have briefly looked into X-ray crystallography and also NMR. So now we will also look into cryo-electron microscopy. There are several steps in cryo-electron microscopy. Firstly, the sample should be purified and structurally intact. Then the sample is frozen by plunge freezing so that it doesn't freeze dry in the microscope's vacuum. This freezing must be very fast because that will eliminate water crystals, which will otherwise affect the structure. So how this is done is that a small amount of the specimen in solution is applied to a transmission electron microscope grid. Then the excess liquid is removed by blotting paper, and the grid is plunged into liquid ethane to wet the specimen and freeze it instantaneously. Then the grid is transferred to an electron microscope, and liquid nitrogen is used to maintain the temperature. After that, imaging is done, and 2D projections of the image are obtained, which are eventually reconstructed by particle aligning and averaging to obtain the basic 3D structure. Finally, there are softwares which can be used to analyze this 3D structure and obtain a final 3D model. Cryo-electron microscopy is gradually becoming more and more important compared to NMR and X-ray diffraction. other than these experimental methods, there are now theoretical or computational methods like molecular dynamic simulations, which help give an idea of the dynamics of the molecule. So the experimental methods like NMR diffraction or cryo-electron microscopy give an idea of this or give a static picture of the molecule. But if we want an idea of how the molecule is moving, molecular dynamics simulation is done. So basically a system of particles is taken. Then for that system, Newton's equation of motion are solved, which helps extract the position and velocity or momentum of each of the particles. And then statistical mechanics is used to extract different properties. In molecular dynamics simulation, usually the time is divided into discrete time steps, and how long each time step should be depends on the time scale of each of the molecular motions. Like you can see, bond stretching happens between picosecond and femtosecond time scale. Elastic vibration of proteins occurs around the nanosecond time scale. This alpha helix and beta hairpin folding occurs around microsecond time scale, whereas protein folding can occur from any time scale between second to millisecond. So using molecular dynamic simulation, a dynamic idea of the flexibility of the molecule can be obtained. So this has basically been a summary class where we have gone through several of the topics that we have learned in different lectures. So that brings us to the end of the live sessions. So I know your exam is closed. Please feel free to contact me with your doubts before the exam. So I'll end the class today. See you later.